Thank you very much for this opportunity to present my work on the energy material science. And we have been using high resolution and high dimensional X-ray tomography to tackle the frontier challenges in this field. My name is Yijin Liu. I'm from Stanford Synchrotron Radiation Light Source. X-ray has been used for imaging ever since it was discovered. In more recent years, thanks to the development of the synchrotron facilities, there are many different X-ray modalities has been developed, has been deployed at the large-scale X-ray facilities. The reason of this is actually very simple because there are so many different imaging modalities. Every one of them has pros and cons. Only if you combine different imaging methods, then you can have a more complete picture of the scientific case that you are looking into. It's my opinion that the tomography is one of the most important development in this field. I think without too much of the effort, I can convince you that knowing the three-dimensional structure of your sample is more informative comparing to only look at two-dimensional projection images. Now let me start to be more specific and talk about this transmission X-ray microscope available at Beamline 62 of Stanford Synchrotron Radiation Light Source. If you look at this schematic drawing of this particular setup, you see that the X-rays comes from the large-scale synchrotron going through the Wiggler, and then it bounces through a number of mirrors before it's focused by the condenser optics down to a few tens of micrometer scale for illumination of the sample. And downstream of the sample, we have a Fresnel zoom plate, which acts as a magnifying lens to project the image to the detector. And that's how we can use the conventional CCD detector to achieve spatial resolution down to tens of nanometer scale. Another major thing about X-ray microscopy with the large-scale synchrotron facility is that you can tune the X-ray energy. Let me give you one example here. Now, in this particular sample, I know that we have iron, we have titanium, we have zinc in this particular particle. Now, if you look at this transmission or the absorption of the elements as a function of energy, you see in this plot that it's slowly changing until you hit a certain energy point where you see a big step. Now, by making use of this big contrast, then we can have elemental specification in our imaging data. As I show in the top images here, we take images above and below the zinc K edge. After the image processing in the differential map on the right-hand side, the only thing remains visible is the element of interest. Now we can do this for different elements. We can do this in different angles. Eventually, we can combine all the information and reconstruct into this three-dimensional distribution of all the elements of interest. Another thing that we can potentially do with the spectral microscopy is to detect the local chemical state of the material. Now, this is a rather busy slide. Let me walk you through this a little bit. As you can see here, we can take images of the sample as we scan the energy with finer steps. Now, if we follow the intensity of a certain pixel in this image, we see that the intensity changes up and down. Now, the shape of this spectra tells us the local chemical state associated with the corresponding pixel. Now, in this particular case, I know that my sample has either nickel 0 or nickel 2+. Then I can use my standard spectra to perform the linear combination fitting. After the fitting, I can dig out a percentage of the fitting results. Then I can assign the pixel with the color selected from the predefined color map. By doing this for all the 4 million pixels in my image, I can get a two-dimensional chemical map showing on the lower left corner. If I combine this with tomography, then I can do this in different angles. Eventually, I can reconstruct the three-dimensional distribution of different chemical species in this material. I should again highlight here that this is full field imaging combined with the spectroscopy scan, so that we can get about 4 million pixel spectra in something like 10 minutes. Now, if you combine this with tomography, then you get a billion spectra, 1K by 1K by 1K pixels in about 10 hours. So there's a lot of information, a lot of data, and waiting for us to extract. I talk a lot about this full field imaging technique. I will also mention a little bit about scanning technique. Now, in this case, I'm showing this instrument available at European Synchrotron, ESRF. In this particular setup, what we do is we focus X-ray to a small spot. And then depending on where we put the sample and what signal we detect, we can have different contrast modalities. For example, if we put sample downstream of the focal point, and we can perform holography, and we can perform phase retrieval to reconstruct the material with sensitivity to some of the low Z elements. If you put a sample at a focal point, and we can collect the fluorescence signal, which give us sensitivity to the elemental distribution. Of course, we can also make use of the coherence of the source to perform coherent-based imaging with higher spatial resolution. So I talk about these different imaging techniques, different imaging modalities, and now I would like to switch the gear a little bit and start to show you the application of all these different imaging methods in the energy material studies. And we have been using these imaging methods to study lithium-ion battery. I'm showing here a schematic drawing of the lithium-ion battery, which is pretty simple. You have cathode, you have anode, everything is soaked in the liquid electrolyte. Upon charging and discharging, the lithium shuttle back and forth inside the electrolyte, while the electrons go back and forth through the external circuit. Now, the cartoon looks cute and simple. However, if you look at the real thing, it's very complicated. Take the cathode as an example. If you image this cathode electrode at different length scales, you see there's structural and chemical complexity all the way from the battery cell level all the way down to atomic level. And using X-rays, we can nicely tackle a broad range of length scales, which is very valuable for the studies in lithium-ion battery. So that's why we have been utilizing all these different instruments available at different beam lines, different facilities to tackle this scientific problem in a synergistic manner. I'm going to show you some specific examples. First of all, to look at the particle assembly in the composite cathode electrode, we have been utilizing this holography or projection microscopy at ESRF. So what we do is we cut a small piece of the cathode and we put it into the beam and conduct a phase contrast tomography on this material. And you see that there are so many particles. They assemble together in a nicely densely packed manner. And the fact that we can resolve so many particles at once give us the possibility to really perform the analysis with statistical meaningful information. And on the lower left side here, I show you a virtual slice through the cathode electrode. The slice near the top shows a lot of red particles, which is labeled as the severely damaged particles. And the slice near the bottom is relatively intact. So that observation of the difference between the degree of damage at the top and at the bottom of the cathode material actually suggests that there's a very severe polarization effect in this battery electrode. If we take a closer look at this, the image, we see that there are many different particles and every particle is unique. And that justifies some of the fine resolution imaging to the particle level. One example is that we utilize the fluorescence mapping capability to look into the spatial distribution of the trace element dopants. And in this particular case, we studied lithium cobalt oxide, which is a dominant cathode material for the portable electronics. And you see that aluminum as a dopant, it is fairly uniformly distributed. On the other hand, titanium naturally segregates onto the grain boundaries and onto the particle surface. And being able to map out the spatial distribution of these trace elements is very important, because as I highlight here, that the doping ratio is only 0.1%. It's very low, so it requires imaging methods with very high sensitivity to really recover the distribution. And this study suggests that these dopants, they have different ways to contribute to the improvement of the performance. Another thing that we have been doing is to look at this individual particle reaction under the operating conditions. And we use the transmission X-ray microscope for doing this. In this particular case, we study, again, the lithium cobalt oxide. We perform the Zen's imaging, meaning taking images at different energies across the absorption edge, so that we have the chemical sensitivity to the different chemical states of the material. And you see that at the very beginning, everything is green at the discharge state. As we charge, it turns red. As we discharge, it comes back green. If we do this relatively fast, over 1 hour charging and 1 hour discharging, you see only 50% of this particle recover to the green phase. If we do this even faster, then you see that the recovery rate is even lower. And this in situ and operando observation of the same particle over long-term cycling really provide the direct observation of the rate dependency in this material. But I should highlight here that the reason that we are able to color map this image is because we have prior knowledge of the sample. We know the chemical fingerprint of the fully charged state and that of the fully discharged state. That's why we can use this data as our prior knowledge to guide our fitting of the experimental data. However, what if we don't have complete prior knowledge? Or what if our some of the chemical species is unanticipated? Then this loop will not work very well. And in those cases, we have to go to the look into the advanced computational developments to use advanced algorithms to perform the unsupervised classification of the data points. Now, in this particular case, we studied this NMC material. We use the classification algorithm to separate all the data points into several different groups based on the similarity of the local spectroscopy fingerprints. And such classification calculation give us a hint about what's going on inside this particle, and it help us to resolve the unanticipated chemical species, which is formed upon aggressive cycling conditions. So I talk about there are many different imaging modalities, and talk about how do we make use of them. I highlight here again that by using a synchrotron facility, we can have multiple of these techniques under the same roof, so that we can really look into different aspects of the material and get a more complete understanding of the sample. Another thing that I briefly mentioned, but I didn't say too much about it, is that the X-rays has excellent penetration capability. That's why we can use our imagination to build different sample cells, to control the sample environment, to look at these materials under realistic operating conditions. And all of that contribute dramatically to the scientific case that we are studying. So with that, I would like to thank my colleagues and also my group at SSRL. And thank you for your attention.